Hello viewers, welcome to our presentation identifying parts of a mosquito larva. My name is Peter Mosili and my colleague is Beatrice Lucky. We are from Tamare Primary School, Meru County, Kenya in Africa.
Introduction. Global Learning and Observation to Benefit the Environment GLOBE program is meant to equip students with necessary knowledge and skills about their immediate environment, their distant environment, and the world at large to environmental degradation, climate change, and how to make the earth a better place to be. Our main objective of this research is to be able to identify different parts of a mosquito larva using the GLOBE Mosquito Habitat Mapper app and a microlens provided to us by the GLOBE program. Our investigation involved identifying a mosquito breeding site and collecting different samples of mosquito larvae, which we used as specimens. All the data we collected was recorded using the GLOBE Mosquito Habitat Mapper app and a microlens and uploaded it using the GLOBE Observer app into the GLOBE website and later used it during our analysis. We studied the head, thorax, and the abdomen and identified the fine details of the larva like tufts, pectin, comb scales, siphon, etc. Even though the schools had been closed for a long period, we were able to adapt to the changing globe since learning continued outside classes using GLOBE Observer app and other GLOBE learning tools. Thanks to GLOBE for supporting and helping us in advancing our knowledge and skills in STEM. The research questions. Are we able to identify different parts of a mosquito larva? Which parts of a mosquito larva do we use to identify different mosquito species? Why should we use mosquito larvae in our research instead of other developmental stages?
Tools and materials we used: mosquito breeding site, a microlens, a phone, and a GLOBE Observer app. Procedure. Step 1. Identify a potential mosquito habitat and take some samples of the mosquito larva. Step 2. Using a microlens, identify parts of the larva by selecting a representative larva for some close-up photos. Use the images on the mosquito habitat mapper to compare and identify the parts. Step 3. Eliminate the breeding habitat. Step 4. Document and use the mosquito habitat mapper to send the information to the GLOBE for knowledge sharing. Step 5. Use the data you have recorded for future analysis.
Results. Major parts of a mosquito larva are head, thorax, abdomen. Siphon. Siphon is an air tube used for breathing. Pecten. Pecten is made up of spine-like structures found along the siphon. Gills. Gills are structures attached to the anal segment. Sandal. Sandal is a part that surrounds the anal segment. Comb scales. Comb scales are tiny pitchfork-shaped scales or scales tapered to a single point found on the edge segment of a larva. Hair tuft. Hair tuft is a small amount of hair growing together in one place on the siphon. Dark plate. A dark plate is a dark patch found on the edge segment.
Discussion. From the first photograph, we observed that a mosquito larva has three main parts. These parts are head, thorax, and abdomen. In addition to that, in photographs A, B, C, D, E, F, and G, it has other finer details which we were able to identify. These details included hair tufts, pectin, comb scales, siphon, gills, dark plate, and sandal. By identifying this part, we were able to differentiate between different mosquito species such as Anopheles, Culex, and Aedes mosquitoes. During the identification process, the mosquito larvae were easy to handle, they were harmless to us, and they had everything we were looking for. This answered all our questions, and therefore our hypothesis was true.
Conclusion. From the above observations, we concluded that we are able to identify different mosquito species such as Anopheles, Culex, and Aedes mosquitoes. The parts and details that are used in identifying different mosquito species include siphon, pectin, tufts, comb scales, gills, and dark plate. A mosquito larva is the best stage for use in identifying different mosquito species because they are friendly, less mobile, and harmless. They also have all the parts and details required for identification.
Recommendations. We recommend that it is safe to control the mosquito during the larva stage since they are harmless at this stage. The methods that can be used include draining stagnant water, covering open water containers, using fish and frogs to feed on the mosquito larvae, etc. All the recorded mosquito data to be properly stored for future analysis and education. Research to be done on what can be done to the mosquito larvae to prevent them from turning into dangerous adult mosquitoes.
Thank you.